Good to have you with me on Planetarian Daily. It's March 27th, 2026. Here's your science and health briefing. Let's start with something that's quietly becoming one of the more important body composition stories in recent years. Researchers have been looking closely at what happens when excess belly fat and low muscle mass show up together. The combination, often called sarcopenic obesity, appears to create a vicious cycle that's more dangerous than either problem on its own. Visceral fat releases inflammatory molecules that directly speed up the breakdown of nearby muscle fibers. At the same time, losing muscle makes it even harder for the body to regulate blood sugar and inflammation, which in turn encourages more fat to accumulate around the organs. This self-reinforcing loop accelerates the kind of systemic damage that shows up as higher overall mortality. What makes this finding particularly useful is how practical the detection can be. Instead of needing expensive imaging or lab tests, simple measurements seem to do a surprisingly good job of flagging risk. A larger waist circumference paired with weaker grip strength or difficulty rising from a chair without using your hands gives clinicians a quick, low-cost way to identify people who might be heading down this path. That matters because the usual focus on overall weight or BMI often misses this specific combination. Someone can look normal on the scale but still carry dangerous visceral fat while quietly losing muscle. My take on this is that it shifts the conversation away from simply losing weight toward a more nuanced goal, preserving or rebuilding muscle while specifically targeting the fat stored deep in the abdomen. The pathways involved respond strongly to adequate protein intake and regular resistance work, so the practical changes don't have to be dramatic. Paying attention to both your waist measurement and functional strength might give you an earlier heads up than waiting for a diagnosis. What small adjustment to your weekly movement or nutrition habits feels most doable after hearing this? Staying with major health risks, the FDA has approved a gene therapy for a rare but devastating inherited immune disorder. Rocket Pharmaceuticals' treatment, Kresladi, is now approved for leukocyte adhesion deficiency type 1, or LAD1. This condition leaves children with almost no ability for white blood cells to exit the bloodstream and reach sites of infection. The result from birth is repeated severe infections, poor wound healing, and often early death without aggressive intervention. The approval is based on a small trial involving 9 patients, yet the outcomes were striking enough to convince regulators. For families who have lived with this diagnosis, a one-time genetic intervention that can potentially correct the underlying defect changes the long-term outlook considerably. It's another reminder that even for conditions affecting very small patient populations, targeted gene therapies continue to make steady clinical progress. These successes are building the technical and regulatory foundation that may one day help far more common diseases. While that marks a clear clinical win for a specific genetic condition, other research is revealing much broader risks that affect far more of us in everyday life. And some of that research, it turns out, may need a careful second look. Scientists are discovering that many measurements of environmental microplastics have likely been overestimated because of contamination from an unexpected source, the very gloves researchers wear in the lab. Plastic particles shed from laboratory gloves appear to be mixing into samples during collection and processing. This means a significant portion of recent studies measuring microplastics in food, drinking water, air, and even human tissues may have been picking up glove-derived debris rather than true environmental contamination. The issue runs through a large and growing body of environmental and health research that has accumulated over the past decade. It doesn't mean microplastics are harmless, but it does mean we've been less certain about exactly how much is out there and where it's coming from than many headlines suggested. Moving forward, researchers will need to adopt stricter contamination controls and blank measurements to account for this laboratory artifact. The discovery is a useful reminder of how even careful scientific work can introduce unexpected variables when we're trying to measure something at such tiny scales. Speaking of environmental contaminants, researchers have found a surprisingly simple and natural way to clean up a different type of pollution. Discarded oyster shells, a plentiful waste product from the seafood industry, can naturally capture and remove rare earth elements from contaminated water. In laboratory tests, the shells effectively bind these metals and transform them into stable mineral crystals that are far easier to collect and manage safely. The method stands out because it's low cost, uses material that would otherwise head to landfill, and avoids many of the harsh chemicals or energy-intensive processes required by traditional remediation techniques. Mining and certain industrial activities release rare earth elements into waterways where they can persist and cause ecological harm. Turning oyster shells into a remediation tool offers an elegant example of circular thinking, one industry's waste becoming another's solution. It's the kind of practical, biologically based approach that feels increasingly necessary as we try to clean up legacy pollution without creating new problems. From cleaning water to protecting ecosystems, another discovery shows how nature's partnerships can be more sophisticated than we realized. In the waters off Hawaii, the Hawaiian bobtail squid depends on Vibrio fischeri bacteria for more than just the bioluminescence that helps it camouflage against moonlight. New research reveals these bacteria are required for the squid's healthy overall development. The squid actively recruits the microbes from surrounding seawater shortly after hatching, and without them, normal developmental processes are disrupted. This symbiosis extends well beyond the light-producing organ. The bacteria appear to influence tissue maturation and other aspects of the squid's growth in ways researchers are only beginning to map. It's a powerful example of how certain microbes become integral to an animal's biology rather than simply helpful visitors. The finding adds to a growing appreciation that symbiotic relationships are often more deeply woven into physiology than we once assumed, with potential parallels in many other species, including our own. That elegant symbiosis between animal and microbe has interesting parallels in agriculture, where scientists are now engineering microbes to support another species under pressure. Researchers created a strain of yeast engineered to produce key nutrients that bees normally get from diverse pollen sources. When colonies were given this yeast as a nutritional supplement, the results were striking. Treated hives produced dramatically more brood, with some trials showing up to 15 times more young bees compared with controls. The work addresses a real and growing problem. Climate change and intensive agriculture have reduced the availability and nutritional quality of natural pollen in many regions. Engineered yeast offers a targeted way to fill that nutritional gap without relying on synthetic chemicals. While it's early days, the approach suggests that thoughtfully designed microbial solutions might help stabilize pollinator populations facing multiple environmental stresses. It's an encouraging example of using biotechnology to support, rather than replace, natural systems. While we engineer solutions for pollinators, nature itself is already shifting in response to changing climates. Research in Costa Rica and Honduras provides the first clear evidence that some tropical birds are reversing their breeding seasons in response to climate shifts. Long-term observations of nesting patterns in these regions show certain species now timing their reproduction in ways that run counter to historical patterns, apparently tracking changes in rainfall and temperature. This challenges previous assumptions. Many scientists had thought tropical birds living in relatively stable environments with less dramatic seasonal swings would be less affected by climate change than their temperate counterparts. The new data suggests that even in the tropics, breeding timing is more flexible than we realized. It adds important information to our understanding of how climate change might reshape ecosystems in unexpected ways, potentially altering food webs and competitive relationships between species. Climate impacts are also driving new approaches to policy. Global leaders who used the En-ROADS Climate Policy Simulator, an interactive modeling tool, came away with a stronger understanding of how different policy levers interact. Participants reported feeling more personally connected to the issue after exploring scenarios themselves, and they were more likely to advocate for policy changes compared with those who learned through traditional lectures or readings. The simulation consistently outperformed passive approaches in building both knowledge and motivation. This suggests that hands-on modeling tools may be more effective at driving real-world climate action than conventional education methods. Giving decision-makers the chance to see the consequences of different choices in real time seems to cut through some of the abstraction that often surrounds climate discussion. It's a reminder that how we communicate complex problems can matter as much as the science itself. From global policy tools to everyday agricultural chemicals, a new analysis reveals a striking gap between different regulatory standards. An examination of Australia's highest volume pesticide products found that 60% of those used in quantities over 100 metric tons per year are banned in the European Union. The study looked at 45 widely used agricultural chemicals through government usage data and highlighted significant differences in how various regions approach pesticide regulation. The gap raises legitimate questions about whether some commonly used products in Australia meet appropriate safety standards for long-term human and environmental health. It also demonstrates how regulatory decisions can vary substantially between jurisdictions that otherwise share similar agricultural needs and economic pressures. These kinds of comparisons are useful because they encourage regulators and the public to ask whether the standards we accept are truly aligned with the latest evidence. Staying with health threats, researchers have developed a promising new route to treat a major tropical parasitic disease. They combined engineered fungal molecules with synthetic chemistry methods to identify potential new treatments for amoebiasis, the intestinal infection caused by Entamoeba histolytica. This parasite is responsible for a large burden of disease in tropical regions, with millions of symptomatic cases each year. The novel drug discovery method merges biological screening with chemical synthesis in an interesting way, potentially yielding compounds that work through mechanisms different from existing antiparasitic drugs. Given the limited treatment options currently available and the growing challenge of drug resistance in many parasites, fresh strategies like this are badly needed. It's an encouraging example of how cross-disciplinary approaches can open new avenues for diseases that have been neglected by mainstream pharmaceutical development. Another screening effort is showing success on a different front, this time in catching cancer earlier. A large-scale program offered lung scans to current and former smokers aged 55 to 74. The campaign detected a substantial number of early-stage lung tumors that would likely have gone unnoticed until much later. Finding cancers at earlier stages generally improves treatment outcomes considerably, often allowing for less invasive interventions and better survival rates. The results add to the evidence supporting targeted screening for high-risk populations. While not everyone in that age group with a smoking history will develop lung cancer, the benefit for those who do can be life-changing. Expanding access to appropriate screening, paired with continued efforts to help people quit smoking, remains one of the more straightforward ways we can reduce cancer mortality in the coming years. Shifting from population-level screening to the tiniest scales, new quantum tools are opening a window into life at the cellular level. Researchers have implanted quantum sensors inside living cells to measure tiny shifts in magnetism and temperature. The technique provides new insights into how efficiently cells handle metabolism under both healthy and diseased conditions. This level of detail at the single-cell scale was previously extremely difficult to achieve with traditional methods. The approach could help scientists better understand the metabolic changes associated with cancer, neurodegeneration, and other conditions. It offers a promising new window into fundamental biological processes that ultimately influence our health. Watching these tools develop is exciting because they let us ask questions about cellular function that were once purely theoretical. Today's selection shows how discoveries continue to span human health, environmental monitoring, and fundamental biology, often in ways that connect more than we might first assume. Before we go, keep an eye on follow-up studies testing targeted nutrition and resistance training specifically for sarcopenic obesity. The early data suggests this dual approach could break the vicious cycle more effectively than focusing on weight loss alone. That's Planetarian Daily for today. If you enjoyed this, a rating or review on Apple Podcasts or Spotify really helps new listeners find the show. I'm Patrick in Vancouver. Thanks for listening, and I'll see you tomorrow. This podcast is curated by Patrick, but generated using AI voice synthesis of my voice using ElevenLabs. The primary reason to do this is I unfortunately don't have the time to be consistent with generating all the content and wanted to focus on creating consistent and regular episodes for all the themes that I enjoy, and I hope others do as well.